Hello and welcome. My name is Ashley Barber, and today I will be presenting my master's thesis entitled Edge Effects on Bird Species Distribution in Forest Habitats of Southeast Queensland. Full details of the research can be read in my final dissertation of the same name, submitted for the award of Master of Science through the University of Southern Queensland in 2018. Today I will briefly cover the topic of edge effects, the human impact on the environment, and the current research and literature in this field. I will then discuss the research question I based my study on, my initial hypothesis, and the importance of the research conducted. I will walk you through the research methodology, including study locations and experimental design, and then the results of the study, and discuss the significance of the findings, comparing them to previous studies, proposing alternative factors that may have contributed to the results, and the future of research in this field. Finally, I will summarize the study and draw some final conclusions. To begin this presentation today, I would like to talk about edge effects and the impact that human population growth and expansion has on forest habitats. As the human population grows, more land and resources are required to house, feed, and support everyone. This leads to habitat destruction and fragmentation. Industrial, residential, agricultural expansion requires land clearing, and then the road networks connecting them all cuts and divides the remaining habitats. Quite simply, every habitat has an edge. These can be natural or man-made. Consider a riverbank, the edge of the ocean, woodlands transitioning into grasslands, or a road running along a field. Previous research has shown mixed results. Some studies show a negative edge effect, others are positive, some showing no discernible effect at all. My research posed the hypothesis that there would be a greater abundance and species richness in the interior of the forest, where they are protected from weather fluctuations, predation, and human influence. It is hoped that this study may contribute to the understanding of edge effects and help to better inform our decisions when progressing human development. 3 study areas were chosen due to their similarities in geographic location and forest type. These were the Mount Cootha Forest, Moggill Conservation Park, and Toohey Forest. All 3 forests are largely dry eucalyptus, with rainforest gullies. 3 locations were selected from each forest to be the study sites. Mount Cootha Forest is in the inner western suburbs of Brisbane, and the 3 sites selected are shown here circled in red: JC Slaughter Falls, Simpsons Falls, and the Gold Mine. An example of the forest types encountered are shown here. On the left we have one of the hiking paths, in the middle is a dry creek bed, and on the right is the undergrowth typical of the forest. Moggill Conservation Park is located in Brisbane's outer west. The study sites are again circled in red, off of Mill Road, Chalcott Road, and Weeraburra Road. Here the images show the diversity of forest types that are found: dense, sparse, wet, and dry. The third forest studied was Toohey Forest, in Brisbane South. The study sites, circled in red, are at the end of Sheville Road, Forest Circuit, and off of Isabella Street. The photo on the left and right show the typical eucalypt forest type, and in the middle is approaching the rainforest-like gully. A comprehensive analysis of each species or genus in the forest is beyond the scope of this study, so a bioindicator was used as a proxy for the ecological balance of the forest. Birds were the focal species for this study due to the vital ecosystem services that they play, and their dispersal ability. Recording abundance shows the total number of individuals observed, while richness is the number of different species identified. At each of the study sites, distinct edges were selected and marked as point 0, or edge. A transect was then run perpendicular to the forest edge. 50 metres in, it was marked again as the mid-interior mark. A further 50 metres in was the deep interior mark. Data was collected at each of these marks, as well as in the open area adjacent to the forest edge, for example the car park or picnic area. The data collected in this study was observational. At each mark, a 5-minute period of complete silence and stillness immediately preceded data collection, in order to eliminate researcher presence on bird behaviour. The data that was collected was observational. As though many birds could be heard, it was difficult to discern their location, as sound can travel great distances. Not only was the number of observations recorded, but species were identified where possible, or defining characteristics were noted for identification later. The results were collated in Excel documents and a two-way ANOVA statistical analysis was performed through the use of SPSS software. The mean abundance of bird species outside of the forest was 28.6. On the edge, it was 36.6, while in the mid-interior, there was an average of 16.6, and the deep interior, 10.6. The data collected showed statistical significance, with a p-value of 0.013 and an f-statistic of 9.923. Similar results were found for species richness, with an average of 11.3 different species observed on the outside, 11.6 on the edge, 6 in the mid-interior, and 4.6 in the deep interior. This was also statistically significant, with a p-value of 0.007 and an f-statistic of 1.943. The data is presented here on a few graphs for visual representation, with an obvious decline from the edge to the interior on both the measure of abundance and richness. So what do these numbers mean? As discussed earlier, every habitat has an edge, and the human impact on the environment is increasing with our population growth. If we can understand how edges affect populations, then we can better manage our own development. This study was localised to 3 forests within the Greater Brisbane area of southeast Queensland, and therefore cannot be directly compared to studies conducted on different regions. However, the experimental design was in keeping with much of the previous research in the field. Previous studies have shown mixed results for edge effects. Some show a positive effect, while others show a negative one, and some show no significant effect at all. The result of this research goes against the initial hypothesis, which proposed that abundance and richness would increase with the protection of the forest in the interior. Undeniably, there are a multitude of other factors that could contribute to species distribution. An important consideration is the significant drought that was occurring during the time of data collection. Each study site was dry. Each creek bed that was seen was completely dry, with the exception of a few small puddles. One possible explanation for the results is that with the drought, resource availability is decreased, and encroaching on the human-occupied areas, such as the picnic areas adjacent to the forest, provides access to food and water. As this study was compiled of observational data, there is of course the possibility of human error. All efforts were made to avoid bias or researcher influence through the identification of study sites prior to entering the field and maintaining silence during the data collection periods. Going forward, studies like this could be improved through a larger sample size, more time spent at each site, as well as more sites visited. Additional time and resources would be required to complete a more comprehensive study that simply wasn't possible within the constraints of this master's research. In conclusion, this research was completed as part of my master's thesis entitled Edge Effects on Bird Species Distribution in Forest Habitats of Southeast Queensland. The data collected shows statistical significance against the initial hypothesis, with abundance and richness decreasing from the edge to the interior rather than increasing. This research contributes to a growing field of edge effect studies. Though the results continue to vary between studies and locations, the importance of understanding how habitat edges affect species distribution cannot be understated. If you have any questions regarding this research or the presentation of it, please direct your correspondence to the email presented below. Thank you.